Hello students, in this video we are going to learn about cell cycle control, which means that how the life cycle of a cell starting from the interphase, mitosis and cytokinesis is controlled by our genetic information, and what will happen if the control does not exist. The things that will happen if the control does not exist is cancer. About 25 years ago, a scientist named Tim Hunt, he discovered through his experiment that the cycle of a cell is controlled by chemicals named cyclin. He discovered that there are cyclin D, E, A, P. Well, where's the C? C basically cyclin that makes the cell not to enter further into mitosis after they are born, after the cytokinesis. So they are actually entering G0. G0 means they will not go further to any cycle of mitosis. So anyway, you can see from Tim Hunt's data here that different cyclin has different peak during different stage of the cycle of the cell. Alright, like for example in S phase, cyclin E has the highest amount, while cyclin A will have the highest amount about during the G2 phase. Alright, now the question is how actually the cyclin do its job in controlling the cell cycle. Now cyclin actually will work together with a chemical called CDK, and CDK stands for cyclin dependent kinases, which is actually an enzyme. And when they bind together, they will activate another target protein. How they activate? By giving them the phosphate. So it's kind of like similar to ATP. When the phosphorus is given, the protein pump will become activated and they have energy to do something. So this way, when cyclin meet with CDK, they will give phosphorus to the target protein, and the target protein will become active. Now, after the cyclin do its job, this cyclin will be destroyed. All right, as you can see from this diagram. All right, different phase of mitosis and life cycle, including G1 as G2, they will have different cyclin. Alright, and then soon after the cyclin do its job, which is activating the protein that will activate certain pace of the life cycle, they will be degraded. So the same thing also happens here. Okay, when they meet with the CDK, they will activate the protein that will cause the S phase to happen. Soon after that, this cyclin is no longer needed and they will be degraded. Alright, there is an interesting fact about the Tim Hunt discovery of cyclin. So after this, you will watch a video. Try to listen to what happened. Did he really try to discover cyclin when he did this experiment, or it's simply a luck that happened to him? Alright, let's watch the video.
I started my own research on sea urchin eggs to see if the same kind of things were happening. I just wanted to get to the bottom of it.
The experiment was very, very simple. That's the important thing. So one day, and it turns out the experiment started at 8 o'clock at night, and the others had all gone out dancing or drinking or something. So I was left all alone in the lab with just me and some sea urchin eggs. And I checked they were fertilized, added the label, and then took out samples roughly every 10 minutes. We just analyzed the samples on something called an SDS polyacrylamide gel, which was very standard and we knew exactly how to do, and do it very beautifully, though I say it myself. I saw this protein, which was actually the very first one whose synthesis you could just detect. And it got stronger and stronger and stronger, and then it faded away. All the other ones just went on getting stronger and stronger. So I looked at this, and this is very extraordinary. And the way the experiment was done, there was no question but this protein going away must mean that it was being degraded, and being degraded specifically. I mean, there was just this one protein went away. All the others behaved as you would have expected.
Looking at the full picture in this way, taking samples over time, let me see that the protein wasn't coming and going randomly. It was coming and going in a very distinct cycle. I looked at this and, gosh, that's absolutely amazing. There it was, on this gel in black and white.
Hunt called the protein cyclin because it came and went with the cell's cycle. He knew that what he was witnessing was significant, but he had no idea what it could mean.
So apparently from the video you found out that it was a serendipity, which means that is due to luck. Tim Han discovers something that is create happiness, unexpected result that is good. And believe it or not, Tim Han actually got a Nobel laureate because of this discovery. So he was not trying to find cyclin. What he did was he tried to to map the protein production from time to time, every 10 minutes, from the embryo of sea urchin that is growing. And he discovered that there are some protein that has a pattern that going up and down, going up and down, and so on. And then he also being observant, because why? Other than just seeing this pattern, he also tried to find out other pattern that might give him a clue. And he came to the pattern of cell division. In this second diagram, you can see that there is a pattern of a cleavage index. If you still remember, cleavage index is when the animal cell divide, okay? There is a cleavage furrow, okay, that will divide the cell into two. Now, he combined the two diagram or the two data and then find out that, oh, maybe these protein are actually the one that control the cell division or mitosis. And apparently, he was right. As what you have learned in the beginning, we will also learn about how if the control system is gone. What happened is the formation of tumor, and which later on will cause cancer. We will talk about what's the difference between tumor and cancer. Now, you must know from now that cancer is not at all a disease that is caused by any pathogens. It's simply because your cell lost its ability to be controlled when it is controlled to be told when it is the time for them to divide and when they have to stop. So as you can see from this picture, there is a tumor that starts to grow here, okay, it's quite large, okay, so it means that there is a medical treatment that has to be done. Now, this is the process of how the tumor starts to appear. So at the beginning, it's just a small lump of cells. As you can see, the cell shape and size of tumor cell is so much different from the surrounding cells. The surrounding cells are actually the healthy cells. So this one are the healthy cells, while this, okay, are not healthy cells. As you can see, they are different, which means that these cancerous cells do not differentiate as the surrounding cell. They are not part of the tissue. That's why they become a problem. Sooner or later, this undifferentiated cell, like the skin cells, they start to become a lump that will invade the space of the healthy cells. They get bigger and bigger, and they will start to become cancer. So what's the difference? When we talk about primary tumor, we talk about tumor or uncontrolled cells division cells that stay in one place, while secondary tumor means they start to spread out. Now, so it means that let's say there was a tumor once that happened in the breast, for example, they can later on, after the lump becomes so big, because they are not tissue, the connection between the cell are pretty loose. As a result, it's easy for the cell to be separated from the lump and carried out by blood circulation to another place. For example, from breast cancer, it goes to the lung and it causes another tumor in the lung. So in that case, it is no longer a primary tumor. It's called secondary tumor. Why? Because the tumor has spread out. Or in daily life, we call it cancer. Now, the process of spreading out the tumor is called metastasis. So we have learned that tumor is caused by uncontrolled cell division. But actually, what caused this control to be gone? Of course, you have learned in the previous part that nucleus that contain genes is the controller of cells. So we can say that the main cause, of course, is the damage that happened in the gene. Now, to make it clear, you must know that there are three classes of genes that involve in the cancer formation. They are oncogenes, tumor suppressor gene, and DNA repair genes. However, lucky you, these two other genes are not part of the DP syllabus. You just will focus on the oncogenes. So let's talk about oncogenes. Before the gene become oncogene, they are called proto-oncogene. So this is the healthy version, unmutated version of oncogenes. Their job is to stimulate cell division in a regulated manner, which means that they control the cell division. Now, proto-oncogenes, they code including the growth factor, factor receptor, and cyclin. So proto-oncogene is genes that codes for cyclin. And when cyclin exists, of course, the cell division will be controlled, as you have seen in the previous slides. Now, however, mutation can happen, and these proto-oncogene become oncogenes, all right? And then this oncogene will cause the cell division become unregulated, uncontrolled. And this is another source that helps you to understand proto-oncogene are genes that normally help cell to grow. When proto-oncogene mutates or changed, there are too many copies of them, for example, they become bad genes, then that can become permanently turned on or activated when it is not supposed to be. As a result, the cell grow out of control and lead to cancer. So again, the most important thing to know is proto-oncogenes mutated, they become oncogene that leads to cancer. So maybe you ask me for the next question, so Ibu, what caused the gene mutation? Now, before we talk about the cause of gene mutation, let's take a look at the detail of what is gene mutation. Now, if you still remember in grade 10, this is DNA, and DNA consists of nucleic acid, and there are 4 different types of nucleic acid. Okay, the ATGC, adenine, cytosine, thymine, and guanine. Now, let's focus on this part. Okay, in this part, you see that there are 2 bases, the adenine and thymine. Okay, this is the healthy DNA that codes for a gene. However, when it is mutated, instead of adenine, it becomes cytosine, and instead of thymine, it becomes guanine. All right, you can see from here. Now, as soon as there is changes in the codes of nucleic acid, we can say that this gene has been mutated. And after many points of mutation, okay, in the proto-oncogenes, this will cause tumor to appear. So please remember, it is not just one mutation, but it is accumulation of mutation that cause the proto-oncogene become oncogene that leads to cancer. Now, let's answer your question, what caused the genes to get mutated? The answer is mutagenic substances. So mutagenic substances are the substances that cause mutation. Now, if the mutation eventually lead to tumor and cancer formation, now it is the substance that is called carcinogenic. The substances that can cause mutation, for example, are radioactive substances. So that's why in some lab that deals with, for example, the power plant that use radioactive as the source of energy, they have to be very, very careful, and they have to have special equipments that protect people from the radiation, because radiation can cause mutation. Now, there is a very famous substance that cause mutation that eventually lead to the cancer. You can guess that this is the cigarette. Yes, even though the data is just a matter of, it's just still correlation. Correlation means there is no, evidence that is solid when you give the concentration of smoke, this concentration, and will cause how much cancer and so on. But there is correlation, means that there is a tendency, a positive tendency, that when people smoke more frequently, the case for cancer to appear is higher. So even though, of course, we cannot test it on human, like ask someone to smoke in certain frequency or certain amount, and then let's check the cancer. But the data comes from, the observation, people who got cancer, lung cancer, and then they are apparently smokers and so on. So there is a correlation. Still, we suggest you to avoid the cigarette, no smoking, because, well, even though it's positive correlation, still it's better to become precautious about this, instead of, after, if you are become a patient of cancer because of cigarette, and then you will regret it. You cannot turn back the time, right? So young people, let's avoid cigarette. So I think that's it from me about the cell cycle, and also the control of the cell cycle, and also the effect or the implication if the control is gone. Thank you so much for your time. I hope you learn a lot. Bye.